All right, so we are live. I'm going to share the slides. Okay. I think everyone can see the slide.
Sorry, Mohammed. Did you communicate with people that you're starting before 10:15? I think usually lectures at 10 are meant at 10:15.
Yeah, that's true. But we already sent the announcement that we'll start at 10.
Okay. Yeah, I can see the slides.
Okay. So, yeah. So we can wait 2 minutes and then see if people will join. Let me record the session. So I'm not sure if if all students prefer to have this course as in person or online. We can have it in person, can have it hybrid if you prefer. But I see some students already send their concern that if it is online, it will be better since they are working, so they can join from work. But if majority of you prefer to have it in person, we can have it hybrid as all other courses. And this is what ETH recommends, to have the hybrid mode. But you can discuss it if you like to have it in person with your mentor. We can accommodate that request. If you like to have it online, that's also fine. Given that all these lectures will be recorded, live streamed on YouTube, and so on. Okay, I think we can start soon. Please, if someone can send me, and if we get any questions on YouTube, that would be great. We can answer them as well. People on YouTube, you can always ask questions in the chat box next to the YouTube video. Students here, they can directly speak up. You don't have to raise hand, you don't have to wait. You can directly interrupt me or us and ask your questions directly. Okay, so let's start. Good morning, everyone. I'm Mohammed Alser, and today is the first lecture in projects and seminars, mobile genomics. So this is our second semester to provide lectures on this topic, as well as project mentorship and so on. So today we'll be talking about an introduction to this course and go through the logistics. And then we will go through the projects that we are proposing to the students so that each student can pick a single project and work on it for the entire semester. Unfortunately, we cannot share the project session, so we're going to disable the live stream on YouTube once we finish the introduction part or the logistics. All right, so what is the role of this course? First of all, we will cover the basics of genome analysis through lectures. So we have 1 hour lecture every week on different topics covering different aspects of genome analysis. This hopefully can enable students to understand the speed accuracy trade-off in using computationally lightweight heuristics. And as you know, heuristics are not optimal. They are near optimal or providing a solution that is close to optimality, but of course, trying to leverage or exploit some speed up through heuristics or heuristic algorithms. And there are other side of the algorithm, which we call them accurate or optimal computation. And these normally computationally expensive algorithms. So it depends really on the application. Sometimes it's okay to use heuristics. Sometimes you really need to use optimal algorithms so that you don't sacrifice any of the end results or the accuracy. Students in this course will experimentally evaluate different heuristic algorithms and observe their effect on the end results. And since this is, as you know from the name of the course, projects and seminars, so they really have to do hands-on experience with some flavor in this course in genome analysis. It could be read mapping, variant calling, and so on, using some hardware accelerators or CPU implementations. So this evaluation will give the students the chance to carry out a hands-on project to implement one or more of these heuristic algorithms in their smartphones and help the society by enabling on-site analysis of genomic data. Of course, we are not going to deal with wet lab analysis, for example, and most of the analyses will be computationally. But we hopefully at the end of the semester will enable having very fast analysis or very fast computational step that will contribute to the overall genome analysis. What are the key objectives of this course? So we are aiming at multiple components that are helping the student to improve first the basic knowledge. So we're going to teach them all the basics in terms of bioinformatics or genome analysis. Of course, as all students are aware, that we don't require students to have any knowledge in bioinformatics, for example. So we're going to provide all these basic knowledge. We'll provide you with the technical skills in genome analysis and computer architecture. So we'll have several mentors for each project, and each of these mentors will be expert in one of the fields or one of the directions needed for that project, in terms of acceleration, algorithm implementation, bioinformatics science, and so on. Hopefully, at the end of the semester, you will have some skills in terms of critical thinking and analysis, especially if you face a problem, and then we will teach you how to handle or tackle that problem very efficiently. So we'll ask you to look for solutions yourself first, and we could provide some suggestions, like guide you in the direction of the correct answer. And once you fail to find a good solution or very efficient solution, we are going to meet weekly with the student, all of us as mentors, with each student, and then guide you directly on how to tackle that problem. So we will never let you handle the problem by yourself and taking very long time to solve it. Of course, that should enable you to get some familiarity with key research directions. So most of the projects that we are proposing are ongoing projects as part of our research. Projects, they could lead to research papers, or they could lead to something else like multiple research projects and so on. But at least you will be really familiar with the environment of doing research. We will have technical presentation of your project at the end of the semester so that you can tell all the people in our research group at Safari, ETH Zürich, what you have been doing and what are the outcomes of your projects, your contributions, and what are the future steps that can be handled in the following semester. What is the key goal of this course? So first, we are going to educate you on how to efficiently implement one of the key steps in genome analysis on portable devices. These portable devices can be anything. So we will leave it to your imagination. We have some real, actual hardware that you can leverage. We already have very efficient and state-of-the-art hardware at the Safari Research Group in terms of GPUs, FPGAs, SIMD processors, processing in memory, near storage, and so on. And some other platforms, we don't have the actual hardware, but we do have simulation platforms. So you can leverage all of these facilities, all of these resources for your benefit. What are the prerequisites of this course? No prior knowledge in bioinformatics is required. A good knowledge in C programming or programming in general. It could be Python, it could be GPU programming in CUDA or FPGA in HLS, and so on. So it depends on the project you pick. You really need to have the programming skills required for completing the project. And you should be self-motivated, and you have the interest in making things efficient and solving problems. So please Don't get bored once you're stuck in a problem, but you have really to be self-motivated so that you can search for solutions and try different methods, see which one is more effective. Even if all of them solving the problem, we are always looking for efficient solution. So this will be our methodology in mentoring you. We can guide you, provide different directions, different methods to be evaluated. And basically you have 1 week, 1 week time to finish some task. And then we meet in the following week. We do follow up on the task that you've been assigned to. And if there are some problems, we can solve it together and then move on to the next tasks. So I think we have about 10 weeks from now to complete the project. Of course, during the last few weeks, you will be having your final exams. So normally we can stop working on projects during last 2 or 1 week so that you can focus on your exams. And then after the exams, you can continue for another 1 week or 2 weeks finalizing your project. We can accommodate all these requests. We can help you handling your time efficiently. And we can discuss all these details, basically. All right, so who we are, basically, I'm Mohammed Alser. I'm lecturer and senior researcher at Sapphire Research Group. This is one of the research group at ETH Zürich, where we are handling hardware, computer architectures, bioinformatics, and many more research directions. Sapphire Research Group is led by Professor Onur Mutlu. I got my PhD from Bilkent University in 2018, and I work at UCLA, TU Dresden, and Petronas. I received IEEE Turkey Doctoral Dissertation Award, and I prefer to use Twitter to share anything related to research. I'm mainly focusing on bioinformatics, computational genomics, metagenomics, and computer architecture. We have, you are very lucky, actually, all of you, to have a large number of mentors in this course. So we start with Juan, who's leading GPU processing and memory research at Sapphire Research Group. He will be part of mentors. We have Noor, who's focusing on bioinformatics and privacy in specific. We have John Fortuna, PhD student working in different flavors in genome analysis. And we have Jeremy, who's a PhD student working on computer architecture and bioinformatics. Joel, who joined us recently as a PhD student. He was doing a lot of research with us before that. And he's working on accelerating bioinformatics applications. We have Max, Bano, and Arvid, who are master's students, and they are doing a good job with us. They've been doing research even before joining our research group as part of the courses. So they were taking a PNS or seminar and computer architecture courses, and then they decided to join us, continuing doing their research. All right, so what are the course requirements? How to success in this course? How to be successful, basically, not in terms of a grade, but actually in terms of implementing the project and finalizing all the tasks assigned to you. So attendance is really required. So we ask all students to attend at least one session every week. And I'm going to give you more details in the next slide. We assign already learning materials in the very first week. So hopefully all of you studied that. We're going to discuss it next after this session. Each student will carry out a hands-on project where you will do the dirty job, or you get your hand dirty with the code, implement, fix, debug a lot of things until you get the project implemented. Of course, that will be with very close engagement from the mentors. And we ask all students as well to participate. Participation is really important so that we can have engagement. We can have an interactive lectures. So feel free to ask questions, comments if something is not clear. And we assign papers normally every week. So you have to read the relevant paper so you got more knowledge, especially those papers related to what you are doing as a project. And at the end of the semester, you'll do a presentation and you will hand your code through GitHub. Of course, we will keep it private until we release the entire project as a research paper. And we will help the projects with good progress, especially those with good contributions, novel contributions, to get published in good venues as conferences or journal. We have very good experience with the past students who participated in research papers, either as ongoing or already published papers. So we can continue doing this practice with you as well if you are doing a really good job. Your responsibilities in this course: we will have a lecture on Tuesday at 10 a.m. Zürich time. We have another lecture on Friday at 9 a.m. All of them will be recorded and live streamed on YouTube. So you need to attend one of them at least. This is mandatory. And you need to work on your projects for about 6 hours a week. This 6 hours can be arranged with your mentor. It could be divided. It could be as one shot in one day. It could be including your meeting with the mentor. So if you are meeting for 1 hour every week, then you can deduct it from the 6 hours and you have 5 hours as hands-on implementation in your project. Meeting with your mentors is really mandatory. Without that, I don't think you can succeed in the project and in the implementation. Again, it's not about the grade, but rather about your benefits and your growth in learning a new topic. We have a website for this course. Feel free to access it. You'll see the slides, whatever we are explaining as videos, slides, everything will be covered over there. Where we will also have a Moodle, especially when we send assignments or homework, so you can hand them easily and conveniently. And all useful information about the course will be either on the website or in Moodle. We will also use the email channel very frequently, so we can send you all the announcement. For each project, we will have a group, which is similar to WhatsApp, but let's say more official between us, so we can see all the progress, details, and that will be for each project. So be only single student and multiple mentors over each channel. Feel free to share whatever you like over this channel. It will be more convenient, I guess, than the email, than the Moodle, and so on. In the next meeting, we'll keep meeting on Tuesday for this session. We will also have another session on Friday, as I mentioned. And once we end the lecture today, we're going to send you an assignment on Moodle on the project's selection. So each student needs to show preferences for multiple projects. And then we will have one-to-one meetings. So all of us as a mentors will meet with every student alone and then discuss the choices he made or she made. And basically, these choices might be good, might not be good. So the students may not be aware about all the required skills needed. That's why we need to discuss it. We will see if you are good in GPU, so we'll do our best to assign a project that requires that skills. If you are good in C or Python or FPGA, we will try to accommodate all your skills so that you feel comfortable working on any project. And it's important that you study the learning material before our next meeting or before you choose a project so that you are aware about all these niches and details in every single project. We will assign the projects next week officially, but after the lecture today, we're going to send the assignment on Moodle so you can choose, but the assignment or the actual assignment will be by next Tuesday when we meet one-to-one. All right, so that's it about the logistics. If you have any questions, I can take it now. Otherwise, I'm going to start with a brief, very brief introduction on genome analysis. Yeah, feel free to use the microphone. If not, you can still ask all questions when we go offline soon after this session. All right, so what is genome analysis? So if you look at this picture, you will see a lot of multiple faces, different characteristics, or what we call phenotype, like skin color, hair color, height, weighing, and all these stuff. So what causes all these differences between human individuals? So that is our genome. And a genome is an organism complete set of genetic instructions. It's really a food recipe where you follow all these instructions and then making you. So you have encoded already in your genome all the instruction needed to make your skin color this way, or hair, body shape, and all this. And this is how it looks like if you try to read it. Of course, the genome or the DNA itself, it's chemical molecules, but we try to convert that as a computer science problem. So we convert it into text. So T is thymine, C is cytosine, A is adenine, and T is thymine, basically. So all these are chemical components or molecules, and we are trying to do some analysis with these data. Now, what we got as a benefits once we analyze our genome, of course, this is unlimited. Just your imagination is the limit here. It could be anything related to our health, to our lifestyle, and everything, basically, not only in Earth, but could be also in space. So understanding the livings around us, like microbiome or viruses, bacteria, affecting our life in every day. And you can see this clearly from the pandemic, for example. So what is the problem here? We expect to do genome analysis by sending a sample to a laboratory, and then they give us the full genome, and that's it, right? Easy. Unfortunately, we don't have any machine that do that job for you. What it does, it gives you really pieces of your genome. And what you do, you send the sample. This is one of the kits available online. You can purchase it for 100 francs or so. And then you send the sample to a company. The company will have some of these machines, which we call them sequencers. Could be really huge, it could be handheld, and so on. These sequencers are chopper, so they will chop your DNA. They cannot process the full DNA as is, and they will give you only pieces of your DNA, as you can see in this picture. Unfortunately, all these machines have something in common, that they lack information about the order of these pieces. You have no idea where each of these pieces is located in your genome. And what we will do, good luck in solving this puzzle. So you have the full picture and you got pieces from the sequencer or the company, and you try to solve this puzzle by having all these pieces and guess whether this is the eye or this is located here, located there, and so on, until you figure it out. And you could do this in a brute force manner. So assuming you have a reference or the reference picture when you solve the puzzle, and you have one piece coming from the machine, and you could check every single location in that reference. Does it match? Does it match? No, move on, until you figure it out. However, this is infeasible. If you want to implement it, it could take ages to get it done. And this is very expensive. And you can see the runtime complexity of this. That is for single piece. Think about many other pieces, that will be a huge workload. Now, how we solve it, assume you have a reference genome, what we call a FASTA file, and assume you have the read set, which we call FASTQ file. And the FASTQ file, the second line only is the genomic information. As you can see here, it's ACGTs. Now, of course, we could have some Ns. Ns here represent undefined. So the sequencing machine cannot decide whether this is A or C or G or T. So it just produces it as N. And we are trying to locate this sequence, the second line in FASTQ file, over the FASTA file, where this piece is coming from my genome. What we do, we use small pieces from the FASTA file, and we're trying to do the same in the FASTQ file. So we just store very small pieces from both files in a hash table or any index. Why we do that? Normally, if you use hash table, it would be very fast to locate any query, basically. If you are searching for a query, the blue query here, the hash table will tell you it exists here and there. But if you search for the full length, you may not find it because there are some differences between you and the reference, especially that the reference is made for a few individuals rather than the entire population in the world. Now, as a matter of fact, if you are looking for something, you expect to have the blue followed by green followed by red as you have it in read sequence. So where we have in the reference genome this exact order, you can see it is around here. So you have the blue piece followed by green followed by red. Of course, there are some characters in between. Then we said, okay, probably our read sequence, by the way, we call this second line as a read. Because this is what we read from your genome sequence. So we said your read is coming probably from this location in the reference genome. And then we investigate the differences between this read and the reference genome. If there are differences in a gene or a specific location, then based on some history, we said, okay, probably you might have this disease or you already have it based on these differences between the read and the reference genome. Of course, it's a bit more complex than what I mentioned. And while you are doing your project, you realize the complexity or the difficulties on finding these locations. That's it in a brief. Now, when we try to do this process in actual implementation, actual algorithm, it's really expensive to do so. So the sequencing machine can analyze 48 genomes in about 2 days. That is one genome in 1 hour. Yeah, exactly. One genome processed in 1 hour, for example. Of course, this is the most capable machine from Illumina, which handle multiple samples in parallel. Now, if we move to the CPU side to try to do the what we call read mapping, which is the previous step I was explaining, we cannot cope with that. It's really expensive, huge workload, such that you can process single human genome in about 32 CPU hours. You cannot cope with the workload coming from the sequencing machine. And this is what we are trying to do in this course. We are trying to solve multiple aspects of genome analysis, starting from improving the bandwidth, improving the energy efficiency or latency, improving the privacy, scalability, and the accuracy of the analysis. You'll see really wide diversity in projects, such that each of them related to one of these aspects. Of course, we are using hardware, we are using software, we are using algorithm developments, or even surveying some existing methods. So you'll see really diversity in the projects such that we can accommodate all your needs. These are the topics we were covering last semester. So feel free to access them, see what kind of flavors we are covering in terms of lectures. And we may improve or add new 2 topics to this schedule as the time comes. So we will send you the announcement, especially the recent work that we publish in conferences. We have some new guests to explain these methods. So we have about 10 lectures, each 1 hour exactly. And these are learning materials to get more about this topic. If you're interested, you can read it on your own. These are our papers, videos, lectures. As I mentioned, all these materials are already on our website, so you can take your time looking at the videos, the slides, and so on. And all the lectures, the material will be online. So now I think the time to move on to the projects. Hopefully we can cover them in 30 minutes. If you don't have questions, we can move on directly to John Fortuna, but we are going to disable the live stream before that. Seems no questions, or we can take it offline, basically. All right. Yeah, thank you so much for people on YouTube. Hopefully next week we will cover more materials. This is just logistics and introduction to the projects, but later on we will try to give teaching materials with the help of members in our group. Thank you so much and take care.